Hello students, welcome to SRM Biology classes. Today's topic is with reference to the main important steps involved in the process of recombinant DNA technology. Main important steps of the process involved in recombinant DNA technology. The recombinant DNA technology, it involves 7 important steps. The 7 important steps are namely, the first step is the isolation of DNA. The second step is fragmentation of DNA. The fragmentation of DNA is performed by using an enzyme called restriction endonuclease. And the next step is the isolation of desired gene by making use of gel electrophoresis. The third important step is the amplification of gene by using polymerized chain reaction. The fourth important step is the ligation of desired gene. Ligation refers to insertion. Ligation of desired gene into the plasmid, or it is also designated as gene splicing. Gene splicing. The next important step is the insertion of recombinant DNA into the host cell. The next important step is the obtaining of foreign gene product. And the last important step is the downstream processing. Are the 7 important steps involved in the recombinant DNA technology. The first step is called the isolation of the DNA, or the isolation of genetic material. With reference to obtaining of human beneficial components, the widely used genetic material is DNA. If we consider a prokaryotic cell, the prokaryotic cell is characterized by the presence of linear DNA. In addition to this, you can find the presence of plasmid DNA. The linear and the plasmid DNA is embedded in a prominent cell wall. This cell wall is characterized by the presence of peptidoglycan, murin and muramic acid derivatives. If we consider an animal cell, the DNA is embedded in a membrane called nuclear membrane. This nuclear membrane is surrounded by a cell membrane. The cell membrane is characterized by the presence of proteins, lipids, and also you can find the presence of carbohydrate derivatives. If we consider a typical plant cell, the plant cell is characterized by the presence of prominent cell wall. In the cytoplasm, you can find the presence of nuclear membrane. The cell wall is characterized by the presence of a chief cementing component called cellulose. This is with reference to the structure of bacteria, animal cell and plant cell. The first step is the isolation of genetic material. Since the DNA is embedded within the nuclear membrane in case of both plant cell and also in case of animal cell. In order to obtain the DNA, we have to break the cell membrane and nuclear membrane in order to obtain DNA. At the time of obtaining the DNA, in addition to the DNA, the RNA, the proteins, in addition to this, the polysaccharides and lipids are separated. The first important step involved in the separation or in the isolation of the genetic material are namely, if we consider a bacteria, in order to isolate the bacterial DNA, the bacteria is treated with an enzyme called lysozyme. The enzyme lysozyme, it effectively breaks the peptidoglycan, murin and muramic acid derivatives, resulting in obtaining of bacterial DNA. In case of animal cell, it is treated with protease enzyme, lipase enzyme and amylase enzyme in order to break the cell membrane. After breaking the cell membrane and nuclear membrane, the DNA is obtained. In order to recover the DNA from the plant cell is treated with the enzyme cellulase. The enzyme cellulase, it will effectively break the cell wall of the plant cell, resulting in obtaining of DNA. In order to recover DNA from the fungal origin, in case of fungi, the chitin is the chief cementing component. It is treated with chitinase enzyme. In order to obtain the DNA. In addition to this, it is treated with several enzymes. Some of the important enzymes are namely amylase. It will mainly break the carbohydrate derivatives. In addition to this, if you observe the plasma membrane or the cell membrane, it is lipophosphorus in nature. You can find the presence of lipid derivatives. In order to break the lipid derivatives, it is treated with lipase enzyme. In addition to this, in the cytoplasm, you can find the presence of various proteinaceous organelles. In order to break the proteins, it is treated with protease enzyme, resulting in obtaining of DNA. The obtained DNA is subjected to ultracentrifugation at 1 lakh revolutions per minute, led to the ready separation of DNA. In addition to this, if you observe the DNA is characterized by the presence of the only protein called histone protein. It is characterized by the presence of two important amino acids. With reference to the histone protein is positively charged. The DNA is negatively charged. In order to exhibit the supercoiling nature of the DNA, it requires the essentiality of positively charged histone protein. In order to remove the histone protein from the DNA, it is treated with protease enzyme. In order to obtain the purest form of DNA. The obtained DNA is subjected to treatment by using chilled ethanol. When it is treated with chilled ethanol or ethyl alcohol, we can readily separate the DNA. At the time of separation of DNA, it looks like a suspension of threads. Such a purest form of DNA is obtained. In addition to this, in order to remove the RNA content, it is treated with ribonuclease enzyme that will readily dissolve the RNA, led to the ready separation of the purest form of DNA. Are the main important steps involved in the isolation and separation of genetic material from different living organisms. In order to separate the DNA from the bacteria, we request lysozyme enzyme. In order to recover DNA from the fungal origin, it requires chitinase enzyme. In addition to this, in order to recover the DNA from the plant cell, we need the essentiality of cellulase enzyme. In addition to this, proteases, lipases, amylases are some of the important enzymes very essential in order to obtain the purest form of DNA. Once the DNA is separated, it is subjected to fragmentation of DNA. The next important step is the fragmentation and isolation of desired gene. With reference to the fragmentation of DNA. The DNA is subjected to fragmentation by making use of a key enzyme called restriction endonucleases. The enzyme restriction endonucleases, it will mainly act on the restriction sequences or restriction sites, resulting in the fragmentation of gene, resulting in the fragmentation of DNA. The obtained fragmented DNA or the obtained fragmented gene is inserted into the plasmid, for cloning. That unique DNA is called passenger DNA. The obtained desired gene is inserted into the plasmid for cloning. That unique DNA is called passenger DNA. After obtaining the desired gene is separated by making use of gel electrophoresis. The gel electrophoresis is a unique methodology widely employed for the recovery of the desired gene. In order to isolate the desired genes from the genetic material, the widely used methodology is called gel electrophoresis. The gel electrophoresis is a unique technique or unique methodology used to separate the DNA fragment based on the size, based on the length, based on the molecular weight, and also by making use of electric charge, the fragmented DNA is separated by using gel electrophoresis. The type of the gel used in the electrophoretic technique is called agarose gel. By making use of agarose gel, the DNA is separated. With reference to the working principle of the gel electrophoresis, the gel electrophoresis is carried by the presence of a semi-solid agarose gel. The semi-solid agarose gel is carried by the presence of meshwork of interlinked linear strands, and it is also characterized by the presence of number of DNA wells. By making use of micropipette, the fragmented DNA is inserted into the DNA well. By making use of micropipette, the fragmented DNA is inserted into the DNA wells. Slowly, it is connected to the electric field. Characteristically, the fragmented DNA is always kept toward the negative charge because the DNA shows negative polarity. Toward the opposite pole, you can find it as a positive charge. When electricity or when electric field is connected to the electrophoresis, based on the molecular weight, based on the size and based on the length, the DNA molecules will start to move toward the positive charge, The smaller size followed by the less molecular weight containing DNA will move very fastly towards the positive charge, whereas the high molecular weight with larger fragments will move very slowly. As a result of the characteristic size and the molecular weight of the DNA, the fragmented DNA is subjected to separation, resulting in the formation of restriction fragment lengthwise. Polymorphism is resulted. Restriction fragment lengthwise polymorphism is resulted. Once the polymorphism is resulted, the separated DNA fragments are visualized by making use of a dye. The widely employed dye in the gel electrophoretic technique is the ethidium bromide. The ethidium bromide, it will mainly stain the DNA, resulting in the clear visibility of fragmented DNA under UV light. Once the dyeing process is done, it is subjected to separation. The characteristic separation of DNA is called gel elution. The gel elution is defined as the separation of DNA. The separation of DNA from the gel electrophoretic technique is called gel elution. After the gel elution, the DNA fragments are purified. By making purified, and they are used for the construction of recombinant DNA technology by making use of a suitable cloning vector. Are the main important steps involved in the gel electrophoresis. The gel electrophoresis is the unique technique or the methodology used to separate the DNA fragment based on the size and based on the molecular weight. In this process, the fragmented DNA is transferred to the DNA wells by making use of micropipette. The DNA wells are always situated toward the negative charge. Later, a mesh-like semi-solid gel is inserted in the gel electrophoresis. The type of the semi-solid gel used is called agarose gel. Slowly, the electric field is connected to the gel electrophoresis. Based on the length, based on the molecular weight, the fragmented DNA will started to move toward the positive charge. Smaller size followed by less molecular weight containing DNA will move very fastly, whereas the larger followed by high molecular weight containing DNA will move very slowly, resulting in the formation of restriction. The restriction DNA is used for the fragmentation. Later, the lengthwise polymorphism is obtained by making use of gel electrophoresis, resulting in obtaining of RFLP, restriction fragment lengthwise polymorphism is resulted. Basically, the DNA is translucent in nature. In order to visualize the DNA is treated with and dye, and the name of the dye is called ethidium bromide. After visualizing under UV light, you can get the clear bands of separated DNA. Once the clear bands of DNA is obtained, it is subjected to separation by making use of gel elution. The gel elution is the unique methodology widely employed for the separation, for the cutting of the DNA bands from the agarose gel. That unique procedure is called gel elution. Once the fragmented and purified DNA is obtained, it is used for the construction of recombinant DNA by joining with a suitable cloning vector. The third important step is called the amplification of gene by using polymerase chain reaction. The amplification of gene is defined as the process of obtaining many or multiple copies of identical DNA or identical gene is called DNA amplification. The DNA amplification is performed by making use of a unique technique called polymerase chain reaction. The concept of polymerase chain reaction was discovered by Mullis in the year 1985. For the discovery of polymerase chain reaction, he was awarded with prestigious Nobel Prize. In case of polymerase chain reaction, by making use of heat stable, thermostable top polymerase enzyme. Top polymerase enzyme is a unique DNA polymerase III enzyme widely employed in order to obtain many or multiple copies of identical gene. That unique process is called DNA amplification. It is defined as the process of obtaining many or multiple copies of identical gene or DNA is called DNA amplification. The type of the technique widely employed in the DNA amplification is called polymerase chain reaction. In case of polymerase chain reaction, the top polymerase enzyme, the enzyme TUC, it is extracted from an extremophile. The name of the extremophile is called Thermophilus aquaticus. From the Thermophilus aquaticus, TUC polymerase enzyme is obtained. It is a type of heat stable DNA polymerase III enzyme. By making use of TUC polymerase enzyme, we can get many or multiple copies of DNA. That many or multiple copies of DNA is used for the recombinant DNA technology in order to insert the gene into the cloning vector. The fourth important step is called the ligation of desired gene into the plasmid. The obtained foreign DNA, the obtained desirable gene is inserted into the plasmid, is introduced into the vector to produce recombinant DNA. The process of insertion of desired gene into the vector to produce recombinant DNA is called gene splicing. The process of insertion of desired gene into the vector to obtain recombinant DNA is called gene splicing. In this process, a suitable plasmid is selected. Plasmid is defined as extracircular double-stranded DNA that will replicate autonomously in the bacteria's cytoplasm. Once the suitable plasmid is selected, it is treated with restriction endonucleases. As a result of restriction endonuclease treatment, resulting in the fragmentation of DNA, resulting in the breaking and opening of plasmid DNA at specific site is resulted. the obtained desired DNA or gene containing sticky ends is inserted into the plasmid. In order to join for the formation of phosphodiester bond, it is treated with DNA ligase enzyme. The action of DNA ligase enzyme resulting in the formation of phosphodiester bond. With the formation of phosphodiester bond, the desired gene is inserted into the vector. That characteristic attachment is performed by making use of a key or molecular glue enzyme called DNA ligase. Such a vector carrying desired gene is called recombinant DNA. In this step, a suitable plasmid is selected. It is treated with restriction endonucleases. It will mainly act on the specific site, resulting in breaking and in opening of plasmid DNA. Once the plasmid DNA is opened, the desired gene is inserted into the plasmid. In order to take part in the formation of recombinant DNA, it requires the essentiality of a key enzyme called DNA ligase. The enzyme DNA ligase is called molecular glue enzyme, molecular stitcher anta karitare. With the formation of phosphodiester bond, the desired gene is incorporated into the plasmid. That unique process is called recombinant DNA. It is defined as the process of insertion of desired gene into the plasmid containing vector to produce recombinant DNA. That unique process is called recombinant DNA. Once the gene splicing is done, the next important step is called the insertion of recombinant DNA into the host cell. In order to incorporate the recombinant DNA into the host cell or into the suitable bacteria, the four important methodologies are widely employed. The first important methodology is calcium chloride and heat treatment. The next step is called gene gun. The next important step is called the use of disarm pathogens. Genes are incorporated by making use of gene gun or biolistic method. By making use of biolistic method, the recombinant DNA is incorporated into the host cell. In order to incorporate recombinant DNA into the desired host, we make use of several techniques. The first important technique is called calcium chloride and heat treatment. In this step, in order to incorporate recombinant DNA into the E. coli bacteria, the E. coli bacteria is first treated with calcium chloride, resulting in the depolarization. As a result of depolarization followed by the dehydration, resulting in the sudden increase in the pore size will be resulted. Immediately, the temperature is slowly raised to 42 degrees Celsius. As a result of increase in the temperature to 40 degrees Celsius, it resulting in the destabilization of the cell membrane. As a result of destabilization of the cell wall followed by the increase in the pore size, the recombinant DNA is slowly entered into the bacterial cytoplasm, thereby the recombinant DNA is slowly entered into the bacteria. In addition to this, some of the other technique widely employed for the insertion of recombinant DNA into the O cells are namely micro-injection, gene gun or biolistic methodology, and the last important one is by making use of disarm pathogens. Namely, the retroviruses are widely used in the disarm pathogen category in order to incorporate recombinant DNA into the host cell. The next important step is called the obtaining a foreign gene product. Once the recombinant DNA is incorporated into the host cell, it is subjected to artificial culturing under laboratory conditions by using culture medium. Culture medium is defined as the nutrients used for growing of microorganisms under laboratory condition under human supervision by providing all the controlled condition. That unique methodology is called culturing. By making use of culture medium, the recombinant DNA containing host organisms are cultured under controlled conditions. Later, as a result of continuous culturing, followed by the very less generative period in case of E. coli, let the rapid multiplication of the cloned cells are resulted. Clone is defined as a group of cells derived from the single cell, resulting in the mass multiplication of both cells under laboratory conditions or resulted by using culture medium. Later, the culture is used for the extraction of desired proteins, vaccines, or obtaining human beneficial components. Later, it is subjected to purification, separation, and it is subjected to marketing by making use of downstream processing. The last important step involved in the recombinant DNA technology is called downstream processing. The downstream processing is defined as the process of recovery of product from bioreactors by using recombinant DNA technique is called downstream process. The process of recovery of human beneficial product from bioreactors by using recombinant DNA technique is called downstream process. The downstream process, it involves the separation, isolation, purification of the products. Later, the obtained product is subjected to series of clinical trials in order to detect any side effects. Later, it is subjected to thorough quality check in order to maintain the purity of the compound. Later, the product is subjected to marketing. Are the main important steps involved in the downstream process. The process of recovery of products from bioreactors obtained through recombinant DNA technique is called downstream processing. Are the 7 important steps involved in the recombinant DNA technique. This is with reference to the main important steps involved in the recombinant DNA technology. By this, it will comes to the end of the chapter. I will post the multiple choice questions once you go through thoroughly and try to answer. Thank you very much. Good luck.